Okay, so if it all is well, I would say that we can go ahead and start. Okay, so first off, good afternoon and a little bit of an introduction. My name is Annie. I see quite a few new faces today, and we're here to talk about the Emerge and Engage trials that led to the approval of aducanumab. And the faculty advisor for this project is Dave Shade, who is online today with us. Let's start with just basically introducing that this was a very controversial approval, and it was in the end approved by the FDA based on a surrogate endpoint, which was amyloid plaque reduction. And what made this case particularly contentious was that this drug was approved against the opinion of the FDA advisory panel. They didn't think that they had sufficient evidence, and yet the drug was approved in 2021, and this sparkled quite a media outcry. Most of the newspapers were negative, and they had negative headlines towards the approval of aducanumab. Some of them were a little bit more neutral, like CNN. Here's cost, size effects, here what's to know. But for the most part, this was a controversial approval and sparkled a negative headline. Let's just move on a quick background of essentially what aducanumab was for. So Alzheimer's disease, as we all know, is a neurodegenerative disease. It is caused by aggregate of amyloid plaques in the brain that causes a neuronal dysfunction. And so the aducanumab is a monoclonal antibody that was designed to target these amyloid plaques and reduce the amount of plaques in the brain. And the rationale behind that was that by reducing the amyloid plaques in the brain, you would slow down the cognitive decline of a person. And this, again, I would emphasize once more that it was approved on a surrogate endpoint. So my first question would just this is really about a chat about this two trials. Is the first question is that I would like to ask the class here is what is the surrogate outcome? Do you guys have an answer? Would you like to just spark a conversation? What's a surrogate outcome?
You use other endpoints rather than the things you want to see, such as biomarkers.
That's a very good answer. Thank you for being the first brave one. And does anyone have anything else to add?
So I didn't hear what she said.
Yes, anyone online? I have a textbook, so. Anyone online or anyone? Yes.
So, surrogate outcomes are used in place of the clinical outcome, and they are typically found on the pathway to the clinical outcome. Like you said, it could be like biomarkers that show up before a clinical outcome is seen.
I like your answer. It's very interesting, both answers. Is there anything else that anyone else want to add to that? If not, we can proceed to the textbook answer that I found. And it's essentially, you both were right. It is a marker and it's an indirect measure that is used in clinical trials as a substitute for direct measures of how a patient feels, functions, or survive. And these indirect measures can be laboratory measures, and most often it is. Let's continue and see what their hypothesis was for both the Emerge and the Engage trials. Their hypothesis was that aducanumab would reduce amyloid plaque accumulation in the brain, and that would result in a slowdown thank you, of a cognitive decline in patients with early stage of Alzheimer's disease. That was the hypothesis for both the trials. So let's take a look at the primary outcome, because this is I think it's very interesting. So their primary outcome is the clinical dementia sum of boxes. So what is that? It's a global scale that assesses the severity of dementia in a person. And what it does, it assesses the severity of dementia across several domains. And it might be memory, orientation, judgment, community affairs, or personal care. And the way it is administered, it is a structured interview. So these are fixed questions that are asked to patients or their caregivers. So it can be either or both. And each domain, so they have a set of questions, and then they rate the answers to each questions. And the sum of these ratings will then be a sum that gets that determines the level of cognitive decline that a person has. And if you see here on the table to your right, and you see that they have a spectrum of severity of dementia, with 0 being just normal. And then they have a 0.5. By the way, remember that number is going to be coming important later. The 0.5 to 4.0 questionable cognitive impairment. And they have this spectrum that goes all the way to 16.0 to 18.0, which is severe dementia at that point. And so some of the examples of the questions are, can the patient remember events or appointments? Is the patient able to manage finances or medications? Or does the patient recognizes faces and location? So remember how we talked just earlier about the surrogate outcome. So I'm going to ask you, is the CDR a clinical or a surrogate outcome? What are your thoughts here? There's no right or wrong answer, really. If anyone online as well as here, what do you guys think of the CDR? Is it a clinical or a surrogate outcome?
CDR would be a clinical outcome.
So you think it's a clinical outcome?
Vicky is the one.
I think it's a clinical outcome in the sense that it's administered to assess someone's cognitive function.
Yes.
However,
Yes.
it could be a surrogate for dementia diagnosis.
Okay. That's the great point there. Anybody else?
People in the sense of the CDR assesses the symptoms of dementia. But in the beginning, you talked about dementia being that accumulation of plaques. And it's a surrogate outcome because it's not measuring the presence of the plaques in the brain, but it's being used as a proxy for determining the severity of disease can tell, or is being used to tell that there's an accumulation of a large amount of plaque.
These are both great points. Is anyone want to add anything?
And I would argue that it's a clinical outcome. It measures the patient's behavior. It's a direct measurement of the patient's behavior. Yes, it's an administered questionnaire, but it's a direct measurement of patient behavior. The second thing is that the association of plaques in the brain with dementia is not proof that they're causative. They could be a consequence of. And so the amyloid hypothesis says that they are causative, but if there's something else going on and they're just the end stage of the disease, then they're not. And we still don't know that. So this is a direct measure of patient behavior. This is therefore, I would argue, a clinical outcome.
Okay. Awesome point, and thank you so much. So, if we can go on, so essentially, there is no right or wrong answer. So Dr. Jobs, I think, is just confirming that is a clinical outcome. This is a clinical outcome, and so we can move on from this and move on to what are the secondary outcomes. Now, for the secondary outcomes, they had more questionnaires. So they had questionnaires like the MMSC, the ADS, the ACDS, and these are all questionnaires that were aimed to assess their neurocognitive function. And then they also add those the PET imaging, SUVR, and the plasma tau levels to see effectively whether the amyloid plaques were being reduced. So to assess both the neurocognitive decline and the reduction of the amyloid plaques through clinical biomarkers. So this was an additional question that Dave and I had thought about, and is what would be a non-surrogate outcome for Alzheimer's? I think we'll probably just answer that, but I don't know if you guys have anything else to add. I don't know if you guys have anything
So
else to add.
I'll just point out that, as Annie mentioned, this CDR is considered a pretty, it's a controversial measure as to whether it's surrogate or clinical. But part of the reason it's controversial is because there are no established perfect non-surrogate markers for Alzheimer's, and nothing is specific to Alzheimer's. The diagnosis of Alzheimer's requires things that are very unlikely to happen. And so distinguishing Alzheimer's from dementia, even other forms of dementia, is often a clinical diagnosis anyway.
All right, great. Thank you, Dave. And we can move on to the methods. So let's talk about the inclusion and the exclusion criteria for the studies, because then I will have a couple of questions that will make us reflect on that as well. So they included patients between the ages of 50 and 85, and all the patients, they needed to have a diagnosis of either MCI, mild cognitive impairment, or early Alzheimer's disease or mild Alzheimer's disease. They all had to have a confirmed amyloid pathology through PET imaging. And remember those questionnaires for the MMSSC, they required a score, I think it was between 24 to 30. And for the clinical dementia rating, so the CDR, obviously the 0.5, which was the questionable cognitive impairment that we saw earlier on the table. For the exclusion criteria, essentially, MRI imaging that posed a risk to the patient or interfered with MRI monitoring was a criteria for exclusion. If they saw microhemorrhages, if they saw cortical infarcts, if they saw white matter disease, that was criteria for exclusion. Any condition posing risk for patients or interfering with the monitoring, again, was a criteria for exclusion, but also anticoagulant, unless you were on low dose aspirate, anticoagulants would be a criteria for exclusion. Now, this is what I think is important. So this is a twofer, let's call it a twofer, two questions in one. And you guys can feel free to elaborate as much as you want here. The broader question is why do we set exclusion and inclusion criteria for a study? So, and then within this realm, what might impact generalizability of a study in this case the most? So you can elaborate. You can start by saying what you think. Why is it helpful to establish inclusion and exclusion criteria in a study? Can we start with that? Anyone? Why do we decide to have inclusion and exclusion criteria in a study? Yes.
In this case, they exclude people with other forms of, like, based on their scans, if they have other visible signs of injury. And that could also relate to different symptoms that could confound and bias the results. If perhaps you can't make the claim that any symptoms are coming from Alzheimer's pathology as opposed to a different sort of disease.
That's a great point. Any other thought?
And I think in this case of amyloid reduction drug is also for safety, because for someone who is on, for example, for someone who is on the blood thinner, it might increase the risk for serious side effects that caused by these amyloid reduction drugs.
That's another great point. Do we have any other suggestion or a broader answer to why we essentially have exclusion inclusion criteria? Yes.
So the inclusion criteria are to ensure that the patients enrolled in the trial have the disease you wish to study. You know, you don't want all dementias, you want Alzheimer's. The second reason for an inclusion criterion is to make sure that the treatment you're likely to use is likely to be beneficial. So for certain cancer trials which target a specific enzyme, we want to make sure everybody has that enzyme defect. And then the reason for exclusion criteria are usually related to safety reasons or to making sure that you don't have things that might look like it but aren't it. And so in this case, the inclusion criteria that they're demented is appropriate because you want mild dementia, that's what you're studying. The inclusion criteria on the PET scan is to make sure that it's Alzheimer's and that there's amyloid accumulation. And also, since this enzyme, since this monoclonal targets amyloid, you want to make sure that it's likely to be beneficial. And the exclusion criteria on anticoagulants has to do with the side effects of the drug, which is the
I can bring it up again. So these are, one thing that I was thinking, like when I was preparing this and discussing also with Dave is like, were there any like exclusion criteria that were impacting generalizability, or something that stood out to me was, the exclusion based on MRI findings, for example, or the medications. Do they stand out to you in any ways, or do you think that there were appropriate exclusion criteria in this case? You partly responded to that.
Yeah, they are. In my opinion, they are, because if the drug causes bleeding or is associated with bleeding, then you do not want to enhance the risk of the drug. And so that's an appropriate exclusion criteria. If the indication is for Alzheimer's, you want the patients to have evidence of, and it treats amyloid plaques, you want patients to have evidence that it's likely to be affected. There are other types of dementia, as you mentioned. If it worked for Alzheimer's, you might try it in other types, but that would be a different study.
If there aren't any other thoughts, then we move on to the general details. So general details of the studies. Well, it was randomized. It was with a treatment assignment 1:1:1. There were 3 groups, and we're going to talk about this very briefly. So there was one group that was assigned to low dose aducanumab, one group that was assigned to high dose aducanumab, and the other group that was assigned to placebo. Now, one important thing about the 2 studies is that they stratified, and they stratified by study site, and they also stratified by apolipoprotein E, which is the genetic carrier. So if you carry ApoE status, they were stratified by that. And what do I mean by that? First of all, if you were an ApoE carrier, you were in the low dose group, you would get a 3 mg instead of a 6 mg. And if you were in the high dose group, you will get a 6 mg instead of a 10 mg. We're going to say that this had changed along the EMERGE trial, but we're going to get there in a second. For the intervention, this essentially was administered intravenously, and the patients were seen every 4 weeks, and they had a total of 20 doses. So the most important thing to remember about this is that in both cases, the EMERGE and the ENGAGE trials, they were both randomized, they were both double masked, placebo-controlled global phase 3 trial, and they were both trying to evaluate the efficacy and safety of aducanumab. Now, one thing is that did I mention earlier that they stratified also by sites, and they conducted this study across 348 sites across 20 countries. So my question at this point is like, do we have any idea as to why that they had so many sites for these studies? Any thoughts of what might have led to having 348 sites?
More size for increase the recruitment rate, get more patients, for sure time.
Yes. Any other thoughts?
Because we would have more patients, it would actually empower to our study.
Yes.
As population.
All very valid, good points. Do we have any other thoughts online? Or any other thoughts?
Well,
So there are
I
two-
would ask, oh, go ahead.
There are two reasons which I see. The first is very pragmatic for the trial. More sites, faster recruitment, and certainly for industry, time is money, and they want to recruit more quickly. The second is there are some countries where you have to have some patients in their country in order for their regulatory body to approve it. And so broadening your reach increases the likelihood of it getting approved in that country. Some countries require a study in the country, but at least some require that some of the patients be in their country. And so both of those, I'm sure you're going to come to the downside of 348 sites soon.
Well, one of the things that I was thinking about, or Dave and I were thinking about, this question, and we were sort of thinking whether in some sites they had a low recruitment, like if they needed more sites because they had at some point like problems recruiting people. So a more pragmatic, more strategic reason rather than just simply, the standard.
I would add, in following on to what Dr. Jab said, I agree, it has a lot to do with money. It's very expensive to have lots of sites that enroll small numbers of participants. And it also, I think, has an impact on the potential impact on the quality of the data and the quality of the execution of the protocol. But in industry-funded trials in particular, I would imagine that those extra costs and those extra burdens are outweighed by their desire to hurry up and get an approved product so they can start making real money, much more money than the trial costs. But for non-industry trials or trials that are not designed to lead to market, kind of increasing marketing, that those extra costs actually become very important also.
Yeah, so most industry now pays per patient enrolled, and so the marginal cost of adding a new site is not zero, but it's not huge. And yes, you do have to do more monitoring, so you have some internal costs. But a lot of industry, bigger industry, big pharma, has in-house people that do this, so they just shift more people from one study to another. Small pharma goes to a CRO, okay, and there's a direct cost for a number of sites. So depending upon which company did this, the marginal cost for adding more sites may not be that large, as opposed to sort of the overall cost of drug development and the fixed cost of their clinical trials unit.
Yeah, I wouldn't disagree. In in the overall scheme of the costs of big pharma trials, these extra costs are relatively small, definitely.
And I agree with Dave. 348 sites, each recruiting one or two patients, is a recipe for very poor protocol adherence.
Makes perfect sense. Let's move on to the next slide. Now we look at both trials. Again, we're looking at similarities, right, between Emerge and Engage. So both trials had the same criteria. So they both enrolled the same age people, the same diagnosis, and the same confirmed amyloid pathology. Now there is, and this is 2 points that are very important. The patient enrollment time, and there were protocol changes, and these protocol changes will be significant in what happens after. And then another important aspect of the Emerge and Engage trial is the futility analysis that was conducted based on pooled data. And then we will see later, because I have slides on this, that led to mixed conclusions and a little bit to inconsistencies there. So while all the initial criteria were the same, then there were protocol changes and futility analysis that kind of like had a little bit of a shift there. In fact, you can see here, there were 2 different protocol changes. So for those who the first protocol changes allowed the people that were having a problem with with diarrhea, the amyloid pathology clearing, to be cleared for their amyloid pathology, and we go back to their initial dosing, so to continue to stay on trial. The second protocol version was established to allow those people with genetic carrier ApoE status to go from the 6 mg to the 10 mg in the high dose. Remember that the ApoE status, they were assigned to the 6 mg initially. And this amendment actually impacted EMERGE more so than ENGAGE. And because they started enrolling patients after the protocol PB4 was approved. So there was a major impact on the ENGAGE trial. But at the end of the day, one key take-home message here, it is that EMERGE met the primary endpoint and did show some statistically significant results in the high dose, and ENGAGE did not meet the primary endpoint at all. So now, Dave and I were talking, and we were kind of like wondering, like, if you have 3 groups, right? How does it work for power and sample size? What thoughts come to mind to you for power and size calculations when you have 3 groups like this, in this case?
Allocation ratio?
I'm sorry?
Allocation ratio?
Yes. So when you have how do you think, another way to ask the question here, it would be, how do you think they calculated the power and sample size with these 3 groups?
Let me put this, I think I know what she's getting at. So you've calculated for 2 treatment group comparison, an alpha of 0.05 and a power of 0.9, right? What are you gonna do in a 3 treatment group? If you want to save the same power of 0.9. You're going to have to up the sample size, because your alpha is going to be adjusted for multiple comparisons.
Right.
And so, so that's what you're going to have to do. Your sample size goes up because your alpha gets adjusted for multiple comparisons, particularly if you're going to compare among all of the pairwise comparisons.
I'm sorry if my question wasn't clear, but yeah, that is the correct answer. In fact, I have the answer right here. So here's what happened. So initially they wanted 90% power, right? So they calculated that they needed 450 participants per group, but they had a higher than expected variance, so they needed to recalculate. And if they wanted to maintain 90% power, they needed more people. So they came up with 535 people per group. So that was the answer that, for the power and sample size calculation. Now, for the analysis, they started, like the primary analysis, they used the mixed effect model, mixed model for repeated measure, the MMRM. They did so for the primary outcome, which was the CDR, and they also did for the other outcomes for the other questionnaires that were analyzed through MMRM with fixed effects. Now, for the biomarker analysis, one thing that's important to note is that the first thing that they did is they brought in a subset of participants for those for that, to establish or to see the relationship between tau levels and PET imaging and cognitive decline. And then they analyzed that subset using still the mixed model for repeated measures and used the correlation analysis. Now, when they mentioned missing data, they assumed that the missing data were missing at random, meaning that they were not influential factors in the participants or the outcomes. And they also did specific sensitivity analysis, and then they did sequential testing. Now, this is important because what they did, to manage the risk of type 1 error rate, right? So they started ranking, they started testing by order on a specified order on ranked endpoints. And so if the high dose was not significant at any endpoint, then testing for both high and low endpoints would stop for all endpoints, for all lower ranked endpoints, I should say. So Dave and I were talking and we were asking, we were hoping to kind of get an answer as to whether ranked testing in this case has helped or hurt. Would anyone have any insights on the rank testing in this case? Is there anybody that wants to talk? Dave, would you like to jump in?
So why, I think another way of thinking about it is if you have only pre-specified one primary analysis and it doesn't work, is there a benefit to pre-specifying some additional analyses in a particular order? And by doesn't work, I mean doesn't show the effect that you imagined. So maybe this is a complicated question. I think it is a complicated question. I think sometimes this is done so that you have an opportunity to pre-specify something that you will consider important if your first choice of something important doesn't turn out to represent what you hoped it would. So in this case, if they first specified the CDR score as their primary outcome, and for whatever reason the study doesn't show a significant outcome for CDR, they have pre-specified other things that they also are saying are important and allows them to present the results in a way that gives them more opportunities to find something meaningful in ways that are somewhat pre-specified. It doesn't replace the primary outcome with some other primary outcome, but it is a way to pre-specify, and that prevents you from being criticized too much for kind of just looking at 1,000 things and finding something that was significant and calling that an important outcome.
What does it do to your alpha?
Well, I mean, I think people that do this are trying to, pretend that it doesn't affect your alpha at all because it's not the pre-specified primary outcome. So you can say, for example, if CDR is not significant here, you could say, well, while our primary outcome was not, significant, the secondary outcome of, and I'm just making this up, MMSE was significant, although it was not our primary outcome. But by pre-specifying it, at least you don't get the criticism that says maybe you looked at 10,000 different outcomes and just told us about number 4,600.
So basically, it enables them to pre-specify the outcomes without adjusting and do several comparisons without adjusting the alpha.
Yeah. Yes.
Sneaky.
All right, thank you so much, Dave, for the explanation here. So let's move on to the results so that we look at, we do have, by the way, a couple of tables that will go back to this concept later. So the demographics really quickly. So the majority of the participants were average 70 years. They were females, they were Caucasian. There was a very low representation of African Americans, very low representation of Latinos, and the majority of the participants were also carried the genetic allele, the ApoE. They were classified as ApoE status, and then the majority of participants also had the 80 or 83% of them were diagnosed with mild cognitive impairment. I apologize. And so futility analysis is what was really complicated because in this study, two assumptions were violated. The first assumption was that both studies will have similar effect, and then we'll see that that's not the case. And the second assumption is that there will be a stable treatment effect over time. But here's where it went really complicated. The authors and the people in the study just said, oh, we declared the study futile because we didn't get that, and the study was halted prematurely. But it was a little more complicated than that in the sense that they initially declared futility, and then there was a shift into a positive interpretation of one trial, and then they went back to their original position and they returned to futility for both trials. And the issue here is complicated because so their analysis was based on, the futility analysis was based on pool data from both eMERGE and ENGAGE. So they pulled them together. But when you look at the results individually, so the results of eMERGE and the results of ENGAGE, you will see that eMERGE has an 18% improvement, where ENGAGE does really not come through for these people. So this is why it violated the assumption that both trials would show similar effects. So in general, and this should be, I promise you, this is not as a complicated question as the previous one, but what is the purpose of futility analysis in clinical trial? Well, why do they do futility analysis? Does anyone want to take a stab at it?
If the drug doesn't work, they want to end the trial as soon as possible, save the money and also. Sorry.
Any
Do
other
you think-
thoughts?
I would like to ask a follow-up question. Do you think those two reasons or rationales always go together?
Together. Do you mean that they always, the futility is always because of both of them, or?
Yeah, I think that's a better way to put it. Is futility analysis always conditioned on both clinical/practical and ethical concerns?
In most of the cases, if the drug doesn't work, then we wouldn't want to involve more participants in the testing trial.
Yeah, so I don't, I haven't worked on many industry-funded trials, so I don't have great perspective on that. But I think in non-industry funded trials, sometimes those can be, I wouldn't say in opposition, but can lead to different conclusions. I have been involved with many trials where we were afraid that external monitoring might suggest doing a futility analysis, might even suggest stopping the trial based on an analysis of a primary outcome measure, even though the investigator group might have thought that the secondary outcomes were still worth obtaining, even if it looked like the primary outcome was not going to show the hypothesized effect. And that in those circumstances, it can be particularly awkward and challenging. And I think that's another reason sometimes to try to pre-specify as part of a futility analysis exactly when and why you might not stop the trial, even if the primary outcome looks like it won't show the intended effect.
That makes sense.
It does. All right, so I had a textbook answer here, but I feel like Dave has come through really with a perfect explanation for it. And the way I look at it is really like answering the question, is it really worth continuing? That's the way I look at it. Let's take a look at, remember like early on when I said that they brought in a subset of participants for the PET composite SUVR and the plasma tau level. And then they had these graphs, and so the first trial, the first panel on left is the eMERGE, and you see that the gray line is the placebo, the blue line is the low dose group and the purple line is the high dose group. And you can see that both the low dose group represented by the blue line and the purple line, so the high dose group, they both show a 71% or, you know, a significant decrease from baseline in amyloid accumulation, right? So whereas the gray line, the placebo line, shows a 4% increase for those who decided to participate in the PET composite SUVR. Now, if you move to the right and you look at the ENGAGE trial, the same thing, you know, it's pretty much similar. Both the high dose group and the low dose group showed not a 71% decrease, but a 59% decrease from baseline in amyloid accumulation through PET composite use SUVR, whereas essentially the placebo line really stayed the same. If we move to the next slide, they did the same for the plasma tau levels. Excuse me. And for the plasma tau levels, you will see the same rationale. So the high dose and the low dose represented respectively by the purple and the blue line show a decrease respectively of 13 and 16%, whereas the placebo line shown by the gray line shows an 8% and 9% increase in both ENGAGE and EMERGE trial. So these were their graphs for the biomarkers for the subset of people they brought in for the biomarkers. Now, I have that table that essentially shows the results of the questionnaire, so the primary outcome, the CDR. And you can see from this table that in the only one that's truly significant is the high dose group in the EMERGE trial with 0.01, and it is barely significant in the low dose group for EMERGE and no significant at all in the ENGAGE trial. And that's for the CDR, okay, so for the clinical dementia rating. Now, for the MMSE, so for the other questionnaire, so the secondary outcomes here, MMSE, ADS, and ADCS, you will see just briefly by looking at it, really that the only one that's showing statistical significance is the high dose in the EMERGE trial, and there is no statistical significance in the ENGAGE trial. So now, Dave and I were talking again, and so we were kind of like wondering, like, why in all of this, why did they do two trials? Why did they do two trials instead of one, say, with like a large sample size, and they decided to do two trials? Do we have any idea?
So guys, this is important. It has to do with the law. So the FDA rules require two adequate and well-controlled studies to approve drugs except in rare disorders. And so the approval process for this, which is a common disorder, required two adequate and well-controlled studies.
Does FDA ever follow?
What?
I feel like FDA always skip something like that.
So it depends. If it's a rare disease, there are ways to bypass it for rare diseases. But Alzheimer's is not a rare disease. Let's look at our Congress.
Doesn't FDA realize that people are using this as a loophole sort of get their drugs approved?
Sorry?
People might actually use this as a loophole to get drugs approved. It's that hasn't ever happened.
People do all sorts of stuff to try to classify things as rare diseases in order to get it approved with one study. But the fact of the matter is that for common diseases, it's two adequate and well-controlled studies.
We actually found much less noble reasons than the law. We actually thought it was a strategic decision, increasing the sample size, combining the data from both trials, mitigating the risk of relying solely on the results of the trial, market pressure, market dominance, the race for being the first to put out a Alzheimer's disease drug, revenue. So it was an interesting, it was interesting actually discussing this with Dave, because I remember telling him I cannot find any noble reasons for these people to trials, but thank you for bringing the law into this. At least it gives me a glimmer of hope that there's something behind this reasoning. So let's go on to the discussion. So essentially, to really wrap it up, is that they had fertility analysis issues. We saw that. One trial showed some some results, I would say, and the other one did not. And then they had study limitations, and even Biogen actually came out and released a statement saying we did not have an adequate representation of all the racial backgrounds in our study. So that was a limitation of the study. So if you were in the advisory panel, or would you have approved this drug, or would you approve this drug today? That's a provocatory question. Would you approve this drug?
So did you discuss safety?
That I think I did somewhere. So, but I think I did somewhere. But I'm happy to go back.
So what was what were the safety concerns about this drug? Remember?
So the safety concern had to do with the side effects, and then the fact that it might have been harmful to people to actually take the drug.
And which ones?
So,
Which ones were of particular concern?
which ones?
Wasn't there a measurable rate of intracerebral hemorrhage with this?
That I don't remember.
So there was, most certainly.
And, intracerebral hemorrhage is typically not a good thing.
Okay. Are there any other thoughts, or so are there any other thoughts, or well, safety is a major concern there. It was a major concern there.
Well, so maybe we should kind of summarize the overall findings, so some mixed results, I think, on some of the primary and secondary outcomes. A figure that we looked at that showed some fairly significant findings on what I think are fairly classified as surrogate outcomes that some people believe are directly causing Alzheimer's, and other people argue that is unproven. And some important safety concerns for a drug, in a drug for a disease that has no meaningful treatment. I think that's what, you'd have to think about if you were trying to decide whether to approve this. Lots of complicating factors.
I suggest you conduct a straw poll, go around the room and ask each person to vote yea or nay.
Require them to do another trial before you make a decision.
So the FDA has often, you can vote nay with a proviso that they might go back and do another trial. That's absolutely. And the FDA has told people to go back and do another trial. So yeah, dead doesn't mean dead forever. It just means dead this time.
No, they did ask for a third trial. But
Yeah,
then
but let's poll the room first.
Can I make a comment also? When you go back to the factors, you see market pressure, and I think that is also one of the major reasons why that happened. Market pressure looks very businessy here, but it was actually people who had Alzheimer's who said, hey, we have nothing else, give us whatever.
Yeah, it's market pressure is maybe something a little different, but you're absolutely right. These decisions are sometimes political, and they're based upon pressure from special interest groups.
Yes.
So anyway, but still, let's have our straw vote.
So maybe just have people raise your hand if you would have approved this.
You would? Yes. So if you vote, so this is, we're the committee, okay, we're not the advisory board. Okay, all in favor of approving this drug, raise your hand.
Wait, my question before. And the drug was shown to increase intracerebral.
Yeah, increased rate of intracerebral hemorrhage. Yeah, I can't remember the exact rates, but.
It's, I think it's in the teens or the low 20s%.
So-
It's
so-
it's a lot, but it's not everyone.
Okay, so vote, who's gonna vote yay?
Well,
Vote to
yes,
approve. So I see 3 hands in the room.
Do a black box one, then?
sure, like knowing what they knew
Yeah, Susan
then.
vote no.
And you can't see, but I'm raising my hand,
Absolutely not.
no.
Absolutely not. Whoops. So, yeah. This was interesting for me, because I think last
But,
year they had a
I-
presentation where I think if I would have said yes, I would have approved this drug last year. So to me, it was important to come to this transition and to know more about this trial so that I could actually have a better decision on whether I would approve it or not. But it was approved and it was priced at $56,000 per year. And ultimately, they ended up withdrawing it from the market in the US in April 2023. But I just felt I just have to really take this optimism. I just felt a sense of discomfort throughout, and Dave knows because we actually had an intense conversation yesterday. I just had this sense of discomfort throughout this whole studying about these 2 trials, and because I don't think honestly that I think it did in the end gave a lot of false hopes to family members, if you think about, their loved ones, So from a patient or family member's perspective, it's also heartbreaking to know that it is really not as effective and potentially harmful.
So I'd like to just make sure everyone understands that the FDA, before they make an approval decision, almost always convenes an advisory panel to give them input from presumably people that are expert in this particular field. And the advisory panel here soundly rejected recommending approval. The vote was very one-sided against approval, and the FDA approved it anyway. And that is remarkably unusual, which is why this was a front page story in the New York Times, that the FDA approved this drug over the advice of its advisory panel. And I can also add, I was working on 2 Alzheimer's trials as this happened. Now, neither of them was primary treatment for Alzheimer's. These were treatment for symptoms of Alzheimer's. And the approval caused us a lot of concern because the having patients in our trials start taking this drug could very well have affected and impacted our ongoing trials in ways that we wouldn't have known. We had no experience with or idea of what the drug might do to our patients in our trial. But the overwhelming opinion of clinicians involved with those 2 trials was, this drug's no good. I would never use it anyway. I doubt it will matter to our trial. And it turns out the clinicians were exactly right, because nobody used it and they took the drug off the market. And I think it's also important to point out, the FDA here did require the drug company to do another trial, but after they approved it. So they approved it with the condition you do another trial. And I suspect, but I don't know, that that additional trial was not likely to show the company or the FDA what they wanted it to show, which might have impacted partly the decision to remove it from the market. And also because there was another drug, a similar drug, same class, that they thought was going to be better, and they didn't want to be competing against themselves.
Yeah, and Dave, if I remember correctly, there was a big backlash from dementia centers across the United States after the approval, with the centers collectively deciding we are not going to use this drug.
Absolutely. I think the clinician response was at the very best completely disinterested in ever using it, and in some cases was openly hostile to the notion that it would be approved after a very, lukewarm, I mean, it really wasn't much of a pair of trials to support the approval and against the advice of the advisory panel. But I have some sympathies for the FDA. This is a bad disease that is quite prevalent and has no known treatment. So I think they've learned lessons in the past that when they just reject treatments that don't look very good, they get in a lot of trouble and take a lot of heat for not offering some hope. And now here's an example where they kind of took a different stance and got the same heat from the other side. That's the
Yes,
job
I
done.
agree. I think this case really tarnished the credibility of the FDA overall, and I do feel that there are still like drugs today post aducanumab. There are still drugs that are generating revenue on the market, and they are without confirmatory trials, and they have the similar accelerated approval pathway situations than aducanumab. So, you kind of wonder whether the lesson has been learned, but that would be for perhaps another session. In the meantime, I think if we're kind of out of time. Do you guys have any other final thoughts? Yes.
I had one thought reading this paper. I know that the inclusion criteria was between 50 and 80, and 50 seemed low to me. That seemed like it was kind of early onset Alzheimer's. I didn't know if that was like similar pathophysiology, or I just didn't know if that was like too broad of an inclusion criteria. Because, I mean, the average was 70, so it wasn't the mean. So it may not have influenced it that much, but I figure if there might be different genetic predispositions for amyloid plaques, then I don't know.
So 50 is a lower limit for making a diagnosis of Alzheimer's. And it's, they tried to get around your concern by making sure that they all had amyloid accumulation, right? So they had to have evidence of what appears to be Alzheimer's with amyloid accumulation. And then they stratified based upon the ApoE1 levels to deal with those who might have a genetic component, an obvious genetic component, a risk factor, and not. So they tried to do everything they could. And, restricted enrollment criteria always limit recruitment. So, and they're always arbitrary. If you say 60 and somebody comes in who's 59 with classic Alzheimer's, you wish you made it 55. So it's just, I don't worry about that. They got basically an Alzheimer's population. But you're right, 50 is very young.
Yes.
I'm new to this clinical trials, Arena, so kindly excuse my naivety. So what I was curious is that when there are doubts about whether the drug is safe or efficacious, what are the laws around phase 4 trials? Can FDA not enforce strict phase 4 trials in order to ensure that the approved drug is not causing damage?
So the FDA does not, although some branches of the FDA can be activist, the FDA typically does not design trials. It says, show us your data and we'll decide yes or no. And they can request a phase 4 study. Usually the phase 4 study is additional safety data, right? And so often it's just a cohort study rather than a real trial.
Right.
Compliance with, Dave, correct me if I'm wrong, but there's an article that looked at compliance at pharma with phase 4 stuff, and it's pitiful. And so it's not typically required, but they don't do a lot of phase 4 stuff, even when it's recommended.
I remember the article, and I remember being disappointed at just how poor the compliance was. I don't remember more details than that, but it's fairly, I think, well understood that when drug companies are told to do something post-approval, they're a lot less motivated than they are pre-approval.
I actually have an insight on that because I'm bringing this exact abstract to APHA, which is what happens to the drug after the FDA approval, the FDA accelerated approval, and how much time do they have to convert, right, to do the confirmatory trial. So from that review that I did, out of 319 drugs, only 7 in the last 3 years have converted. So that means that you have all these drugs on the market that stay on the market until at some point the FDA is gonna say, hey, you know, are you guys going to convert at any point? You can present, because the FDA can essentially say, you know what, if you guys don't convert, I'm just going to withdraw the approval. But it's really, really hard. I mean, it's really like hard that the FDA really steps up and does something like that. It's really, there was a new regulatory framework that was the FDORA of 2022 that has institutionalized a committee to oversee all these trials that are essentially, that all these drugs are essentially approved through accelerated approval. And then they should be required to have a confirmatory trials, right, in a timely manner. But it really, there is no specified time of what a timely manner is. So it's really, there's a lot of ambiguity still there. So it's still a hot topic and it's still going on even post aducanumab.
Thank you.
Does that answer your question, right? I think we're running out of time, guys, although this was really interesting. Thank you so much for coming today.
And thank you, Annie.
Any variations, combinations, permutations on your title?
Like I wouldn't have to talk about it, and I want to make sure that I capture that. That's last week.
Yeah.
Yeah, that's before the meeting.
Good job. Good job. What about you?
It was really.
Yeah, so I guess I felt positive having a really precise.
Right.
Yeah, it's only wanting to stuff precise. And since you're an NHS and stats, you should do that.
And then.
I don't know a good term for it.
Right, and then you. I'm such quality. Started over here. And then I asked Ian on this call, when to.
Yes.
I used to see you in class or last year.
You want to do tools? I don't know what tools, I don't know, so Ian and Ann, great.
Yeah.
And, so, and, yeah, I didn't know what to call it, so you might ask, Ian, if you've got the name for a phase 2 trial that needs to which is not replicated in phase 3.
Right, yeah, and then Ian kind of is why are we focusing on oncology, because it's a methodology project and it should be more in these areas and stuff.
Because you have to choose something.
So it's like I already got like 10,000 records just focused on oncology.
I think it's a subset, it's a quasi subset. Can't choose out the genius, but at least it's
Something.
done. You know, and the other problem is that very often in a cancer trial, you're using some of the same.
We're gonna be setting up
Right.
for our
Especially
clearance.
with the same face.
Excuse me, we're gonna be setting up for our
So in phase 2, you're looking at tumor, right?
All right, now, we get, oh, this is, what's this? I'll close these. Okay, all right. Sounds good. I don't mean to be too aggressive, but. No, it's okay. How was your commute to campus?